as I sat through this meeting, I kept wondering about the natural history of a tumor, about the way a tumor changes when it starts to when it is a fully developed and potentially metastatic creature, and the stromal environment around it. So we can start with a fairly simple question germane to that. Does anyone know what the concentration of glutamine is in the tumor environment as you go through the natural history of a tumor?
So the answer actually has been well published in the 50s and early 60s by Dr. Roberts, who had his entire career at the City of Hope. And he spent a time doing animal models of xenografts as well as human biopsies to show that, in fact, he was interested in the same problem that I sort of introduced. The whole problem is, how do cancer cells get enough nutrients to fuel their division? And he thought the limiting thing would be amino acids. And he persistently showed in every human tumor and in every xenograft that the limiting amino acid at the time you could feel a lump was glutamine. And that became his whole career, that we should pay attention to glutamine. It actually launched, as Chi would tell us, the first drugs to actually attack the glutamine synthetic pathway, which unfortunately all were glutamate analogs and ended up having either psychiatric or seizures as a side effect, and it got abandoned in the early 1990s after 30 years of drug development.
So a follow-up to that would be that you would predict that as a tumor develops, there would be an increased requirement for glutamine, and the level of glutamine in the environment should go up.
So, and I think,
do we know that?
Well, so Josh Rabinowitz just completed a study as part of the Stand Up to Cancer grant that Josh and I were part of, which was to study pancreatic cancer, where the study was to get 50 serial patients operated on to have a complete pancreatectomy because they were considered a candidate for a surgical cure or potential surgical cure. As you know, about 10% of patients can actually be subjected to a Whipple procedure and have an increased chance of living. And in that, the pancreas is taken out as a block. And so it was done in the OR with a technician present right there. The tumor was handed off at Chi's Cancer Center by Jeff Drebin, who's the head of surgery there, who did personally every one of the surgeries. Immediately flash froze normal pancreas and immediately flash froze the tumor. And then Josh Rabinowitz did a complete metabolomic analysis and was able to document in much greater detail than you could do in the 1950s exactly what Dr. Roberts found, which is the 4 or 5 amino acids, depending on how you count them, that are critically dependent on glutamine, and glutamine itself are the most limiting nutrients that could be found in the tumor environment. And after that, glucose was the most limiting thing in the tumor environment. And that's the entire tumor environment, and people need to understand that.
I object to the notion of calling cancer a thing.
I agree.
And identifying it as something that we can describe, because cancer is many things. And if we were talking right now about prostate cancer, we wouldn't even be having this discussion. So it behaves so differently from the model we just discussed.
So how well do we integrate the things we've learned today into that particular fact? Because it's clearly true. Cancer's 100 different diseases, 500, I don't know how many, right? And are we to assume that there's a one-size-fits-all metabolic description of them? You're shaking your head no.
I don't think so, no.
No, definitely not.
So what do we know about the types of cancer that are most relevant to the sorts of things we've been talking about at this meeting?
I would say the following. I think we don't really fundamentally understand as a field the cell biology, the metabolism of cell growth in net, even when you do it with tissue culture cells, with the artificial media we all share. Until we understand the rules of that, we are not going to be able to deconvolute that the prostate has a special role that is adapted to produce citrate so that sperm don't waste their ATP load until they're actually freed to actually reproduce the species, which is why the citrate is such a special part of a prostate epithelial cell's biology. We won't understand why glutamate is an absolutely special nutrient in the environment of the brain, because in fact to do neurotransmission you have to have the glial cells scavenge all the glutamate out of the body and not have it available in any way in the extracellular environment, or you'll screw up synaptic transmission. And so there's a fundamental biological paradigm in which the neurons and the glial cells engage in a dance in which they mutually reinforce specialized metabolism. we're going to have to learn all those to understand all 400 cancers. But if we don't start with understanding what fuels anabolic growth, we'll never get anywhere.
Maybe if I could follow up on that. I think Craig's making a good point about the environment and the state of differentiation of a tumor, or the state of differentiation at the time that a tumor begins to occur. And we know, for example, that KRAS, for example, will give colorectal tumors, pancreatic tumors, and lung tumors. Yet if you take a normal lung and a KRAS mutant lung tumor, or a normal pancreas and a KRAS mutant pancreatic tumor, or a normal colon and a KRAS mutant colorectal tumor, and do mass spec metabolomics on each of those 6 things I just described, you would find, what we thought we would find is that all the KRAS ones would have something in common compared to the normal tissue. But in fact, in every case, metabolomics of the KRAS lung tumor looks more like lung than it looks like KRAS colorectal or pancreatic, and vice versa. So I guess the point I'm getting to is that as a tumor begins to evolve, it's starting out at some level of differentiation and in a particular environment. And it has to figure out how to survive and grow in that environment. And the environment of every tissue is very different. So different tumors will take a completely different path. And even each lung tumor, even each KRAS mutant lung tumor isn't going to take exactly the same path, even though they have KRAS probably as one of the earliest events.
What does that say about the relevance of cell culture models for these
Well,
diseases?
yeah. Obviously, cell culture models are disastrous. I mean, for things like prostate cancer, there are only 6 prostate cancer cell lines out there after thousands of attempts to make them. Who knows how many tens of thousands of attempts, 6 successes. And what that tells you is that those 6 can't possibly represent the disease. They represent 1/10,000th of the disease. It's such a rare event to get such a cell line. So I think that this thing with organoids is more promising because you can take it straight out of the tissue and try to replicate the environment of the tissue. Human into mice PDXs, they have some advantage, but again, it's you're missing the whole immune system complexity in that. We have no good models that any of which quite mimic the environment the tumor has in us, other than us. And so in some ways, doing the experiments in humans is the only way we really know the answer.
Would any of the speakers in the audience, right? The first row is other speakers. If you want to jump in on this, please just raise your hand and let me know, okay? I think we can start taking a question or two, but first we'll give you a chance to weigh in on this.
So addressing the question there, which I think is most of you, the panel, are actually talking about established cancers from what I hear, and not for prevention of cancer. There is incredibly strong experimental and human evidence that the environment plays a very important role in prevention of cancer. And probably best typified, as I mentioned in my talk yesterday, by the migration studies from one population to another. So certainly there is something in the environment that can change or prevent cancer. And the evidence is far, far strongest that it is diet, basically from experimental models, which show that there are certain dietary things that can induce cancer. And obviously my talk is more sort of biased towards colon cancer, where you have definite molecules that have been shown to actually promote or suppress cancer, such as butyrate or secondary bile acids, which are carcinogenic in experimental models. So I think from the viewpoint of colon cancer, where you get differences of 100-fold between different populations, it's not an intrinsic genetic difference between the different populations. It's environmental and it's dietary.
You had your hand up way in the back, and I see you. We'll get to
Yes,
you.
2 questions. One, as a medical oncologist, one of the common practices in the supportive care of cancer is the use of many grams of glutamine in very high doses. And I think, having learned about glutamine addiction, I've often cautioned my colleagues about the unintended consequences of extremely high doses, and if anybody might comment on that.
I think somebody on the panel ought to weigh in on that. Would any of you care to? No free medical advice
I
here?
mean, up to 45 grams a day. I mean,
So-
we're talking extremely high doses.
So the biggest problem with giving anyone advice, and Memorial Sloan-Kettering, where I work, is we employ a large number of dietitians who give advice, and we know that our patients take more pills that they buy from GNC, including glutamine, than we actually give them. The problem with glutamine is, and this is for those that are coming from the metabolic disease community, we know that glutamine is just as exquisitely regulated in our extracellular fluids as glucose is. You will catabolize every muscle to non-existent for the liver to have the ability to do glutamine generation that provides a 0.5 millimolar concentration in your entire fluids of glutamine. And so we also don't know, because there are good studies that have been done, how much diet really affects that fluctuation. So it's pretty hard, even by the kind of dietary supplements, to really defeat the liver's ability to maintain a constant, because it'll also absorb glutamine in. And we can see that actually since we've made a fluorine 18 modified glutamine, so we can start to do PET imaging of glutamine. The liver will take it up if it becomes an excess. It's regulated just like glucose. But we don't have any clue, I would say as a community, what the hormonal axes are that are regulating that glutamine. So you're up against that barrier.
And maybe Luke Cantley can address this, the differential effect of fructose versus glucose in tumors, particularly in transketolase and pancreatic cancers, and whether that is a target we should be looking at in terms of diet.
You should be up here. You're asking all these tricky questions.
Yeah, so first of all, just to make the point that maybe everybody in the audience doesn't know this, hopefully this audience, maybe everybody does know, that fructose can be in certain tissues, mainly in the liver, be metabolized through fructokinase rather than hexokinase. In fact, the liver has gone to some trouble not to express a hexokinase. It uses glucokinase for glucose and fructokinase for fructose, and that allows it to escape the citrate ATP negative feedback loop. So you can, no matter how much fructose you eat, your liver never gets full of it. It'll keep making more and more citrate. It'll never shut it off, which is why fructose is such a great way to gain weight. It all goes into fat, and there's no feedback to say quit eating it, and the level in the bloodstream never gets high enough because the liver clears it all. So it's called a low glycemic food. And so people say, we used to think it was great to have fructose because there's no glycemic index overshoot, no insulin response, except after about 2 or 3 months of eating huge amounts of it, you're insulin resistant because of fatty liver. And so, and if you take it with glucose, as we all do, that glucose now is going to be intolerant. Now, but that's not your question. Your question is, does fructose ever get used as a preferential fuel for some type of cancer? And we've looked at that a little bit in some cell lines. In fact, Costas, postdoc in my lab, is probably in the audience who knows in some detail, if he's still here, what cell lines he's looked at. And we just have not seen any that significantly use fructose nearly as much as glucose. So I think you might think that hepatocellular carcinomas might, but most genes that are specific to the liver are lost whenever you transform them to a hepatocellular carcinoma. So I would bet even hepatocellular carcinomas don't have fructokinase, but maybe somebody in the audience knows that.
Is there any future with regards to using diet and bioactive nutrients, whether it's phytochemicals, plant extracts, omega-3 fats, with standard therapies to enhance the effectiveness of the standard therapies in order to, say, sensitize the cells to those standard therapies, because these things obviously affect these pathways that appear to be beneficial?
Which one of you wants to take a stab at that?
I'll make one stab. I think that I think we've not used the right animal models to address these questions. We haven't done it well enough yet. I've seen a few papers that have looked at chemotherapy, effective nutrients on chemotherapy. And the result, the paper that sticks in my head was pretty striking. where I think there were either 2 or 3 different cell lines. It was human cell lines. I believe they were breast cancer cell lines put into a mouse as a xenograft. And then the mouse was either put on a high-fat diet, a regular chow, which is also high sugar, of course. What we call high-fat diet for mouse is really a high sugar and fat, or a ketogenic diet, which is a true high-fat diet with no carbohydrate. And then the mouse was given chemotherapy. Actually, there's a 4th arm of that trial in which the mice were starved for 24 hours prior to the chemotherapy. And then they did all the right things. They looked at C-peptide and glucose and insulin and amino acids, everything in the serum, and then asked what happened. Did the diet affect the response to chemotherapy? And the result was quite striking. In one cell line, the diet had 0 effect on chemotherapy. The same amount of cells were killed no matter what diet it was. And on the other cell line, it had a dramatic effect in that the high-fat diet, where insulin levels were high, completely protected the tumor from chemotherapy, while the starvation diet and the ketogenic diet dramatically sensitized to chemotherapy. So unfortunately, they did not look at mutations in the cell lines, and they didn't ask whether the insulin receptor was expressed on the cell line that showed it was protected by the high-fat diet. I'll bet that's what was happening. But I think these are questions that have not been properly addressed.
Go ahead.
If I just can address the very difficult question, is cancer preventable? I'd like to introduce maybe a concept. Even when we know what could prevent cancer, sometimes you can't, because for breast cancer, you can't say you have to have a first child before age 20, you have to have 5 children, and you have to lactate all of them for 3 years. This is clearly a way to prevent breast cancer, but the society will not accept it. So this is why we need you, because we may need ways to prevent cancer that are medically and acceptable and socially acceptable.
But as far as the diet is concerned, the kind of diet that people usually eat, across the globe, is so varied. And do you think we can test this in a mouse model? I mean, this mouse lives for only a year and a half or 2.
David Sabatini's done some interesting studies, and others have as well, in which one puts a mouse, engineers, or either transplants or engineers a genetic model of cancer. One of them was a lymphoma model, a MYC-driven lymphoma model. In fact, maybe Mike might know more about this than I do, in which the mice were then put on various diets. And there, a low-calorie diet definitely suppressed the growth of the tumor. The mechanism by which it suppressed it wasn't very clear. On the other hand, exercise, which managed to lower glucose levels similar to the restricted diet, calorie diet, was not as effective as the low-calorie diet in that model. Am I getting it all right, Mike? Do you remember this model? Yeah. So, those kinds of studies have been done. One advantage of the mouse is its metabolism is so much faster than our metabolism that things just happen faster. So the mouse, obviously, it's only living 2.5 years, and yet you get a lot of cancers in that period of time. And so that's, that may be, in fact, because their metabolism is so much faster that things just happen. Mutations happen. Although if you look at the cancers in mice, they typically have fewer mutations than human cancer, presumably because they haven't divided as many times. So it's, I think we learned something from it. But.
Michael?
Yeah, so just to give you an example, you may find this reassuring or totally disillusioning, I'm not sure which. In the April issue of Cell Metabolism, there's a report of a study with a large number of mice, fed 25 different diets, where the aim was to modify the components, so as fat, carbohydrate, protein. The first conclusion was calories per se were irrelevant. Now the test was is my problem with this. It was a question of how well would an established tumor grow in these mice, as I recall. That was the test, I think. But anyway, that's the test they used, and there were profound effects. They haven't made the carbohydrate fans very happy because the calories per se were irrelevant. It was the blend of components that was important, and the most offensive were proteins. These papers also attempted to implicate, to explain the protein effect on branch chain amino acids. That they were alleged to be the culprits.
Can I just make a comment regarding my nihilism with regard to mouse models? First of all, the C57 Black 6, which is the most common mouse model, is used. These mice run around with blood glucoses between 100 and 200. You put them on a fast, their blood glucose drops in half. This is not what people do. They have no brain. I mean, they really, so they're
Well,
basically.
neither do many people.
And I don't accept myself in that. But the thing is that, nonetheless, that gives me the opportunity to comment about my mouse colleagues. And the thing about them is.
Hey, I was department chair.
Is that they have no need for ketosis because they have no brain. And if you try to put a mouse in ketosis, for example, they might develop 4 times baseline, whereas a human will develop 20 to 35. So metabolically, they're just quite different than we are. Furthermore, well, anyway, I think that's just the beginning.
Hey, you've been waiting patiently. Please go ahead.
Okay, so the advice for cancer patients from agencies like the WCRF and the American Cancer Society is that they should follow the recommendations for cancer prevention. But presumably, because, say, for example, an obese woman gets breast cancer because she's obese, could it be that that cancer has developed in a metabolic situation that might actually set it on a trajectory that's always going to have a poor prognosis? And whatever she does afterwards, is that going to potentially modify that outcome? Because I think that's the sort of the message that's being given out. And I guess it's probably, it's going to be variable, but...
I think that's a wonderful question. Who'd like to take a stab at it?
I'll just say one thing. I mean, this country now has is going to 16 million cancer survivors. And that population is the single identifiable group in the population at highest risk for cancer. And so they're all tied to the oncology community. And it's actually a shame that no one is actually studying exactly the issues that you're talking about. They're all seeing nutritionists and cancer centers that are represented broadly here. And it's something that has to be systematically studied because they all want to do something to prevent their prevention. So they'll do the screening tests. But it would be interesting if we had good nutritional studies that started to address these issues. So I would challenge the whole community, just as you have. There are dozens of studies on cancer survivors, but not with specific supplements. What does folate do? We've had this wonderful experiment that's been done in England where we've seen neural tube closures go back, but nobody has secondarily analyzed that. But almost all the studies argue that if you want to improve cancer cell differentiation, we are all sub-effective for folate levels. You could ask for that. Does the glutamine that somebody's asked been tested? I don't have a clue. There's not a rational study done in the way Michael just told us about for metformin about the use of unsaturated fatty acids or saturated fatty acids in that community. That's what I think that's what she's challenging us to think about.
So I would just say, if I could have a process as well, that again, something we started out with, that every cancer is different. I mean, there are thousands of different cancers. They've all evolved to survive in different environments. So what's very good for protecting from one cancer may in fact be the worst possible for another cancer. So I think this is not a question we can ask globally. It really has to get down to molecular mechanisms and understand based on the mutational events and tissue of origin of that cancer.
Well, let's see. We've got a pretty good representative sample here. How many of the panel are on low carbohydrate diets? 2. How many of you are on low fat diets? Low protein diets? High protein diets?
Moderation.
Low iron diets? And you in the back.
Do we know whether the mutational landscape of tumors from obese people is different in some ways to the mutational landscape of tumors from lean people? And if not, are the big tumor sequencing projects going to try to make inroads into that question?
Yeah, I can address this a little bit. I think the quick answer is I don't think it's known, but Mike Stratton, we just heard a nice talk from him. Actually, Mike and I did at a meeting we were at last month, in which he's now teased out sequence mutational events, the sequence around particular mutational events across now about 10,000 tumors, and it breaks these down into certain subsets of patterns. And some of those patterns can already be interpreted as being due to UV light versus chemical carcinogens, et cetera. So I think it is going to be possible to tease out if you but you need a lot of data. You need see a lot of mutations in a given tumor, but most tumors do have a lot of mutations to tease out what pattern is there, and that will probably predict what the event was that was causing the tumor. I would suspect there would be, the Fenton reaction, free radical hydroxy. We'll know what kind of mutations that's going to show up, and that'll tell us whether those were the mutational events that was driving that tumor.
Okay. I've got a question for Steve, because one of the things that came up throughout the course of the meeting was the importance of thinking about combination therapy for cancer, that there's not only is there no one size fits all, but there isn't going to be a single treatment that's likely to be effective for any cancer. And one of the things I heard at this meeting that seemed to me to be ideally suited for combination therapy of some sort was Steve's ACSS2 compounds. And I'm wondering whether he's thought about that and how he might go about trying to figure out what would be an appropriate combination therapy in that case.
Well, I think it's safe to predict that any new therapeutic is going to be used in combination. So it's way too far ahead of me to have thought what might be the right partner for an inhibitor of ACSS2. It's not even in the clinic yet, so it's way ahead of us to think about that. But in the context of prevention, I think what you would like for a preventative medicine is a medicine that's fully safe, that has no effect on the patient whatsoever. So a woman has breast cancer and has treatment, and as I understand, they go on adjuvant therapy. Is that right? Thereafter?
Yeah, that's right.
With a toxin. And so I think if you could come up with a non-toxic adjuvant, that would be of potential utility.
I think we need to begin to wrap things up.
Greg?
So I'm going to ask each member of the panel, starting with Mike, if they want to make any final closing remark. Michael?
Yeah, there's a principle's been unsaid here. It's obvious. The best way to design a prevention is to know the cause. And the sorts of studies we talk about of diet and what have you, they may or may not relate directly to cause. They may represent a way to mitigate the effect of a cause. So I think we really need to know the cause of each cancer to effectively design a prevention. We're going to be very lucky to absolutely eradicate a cancer, as we're coming close to with cervical cancer, someday lung cancer, and that's happening because we know the cause. And it's a problem it's also a problem of population biology by and large. Experiment has not helped us a great deal until we have clues, and then even then we don't trust the animal models very well. So I think we need to blend into discussions of how we prevent cancer when we don't know the cause into how do we determine the cause of cancer from population data and however else. It's truly, I think, the most difficult form of cancer research, and it is still to this day neglected.
Well, actually, this may not be the moment to raise speculations of this kind, but I'm going to do it anyway. one thing, the associations that we are aware of obesity, and we did discuss the links of obesity to hyperinsulinemia and hyperglycemia. If we're to look at the last 100 years, and particularly the last 30, and what's happened to our diet, the other feature that's important about this is that mechanistically you say, well, okay, well, what's this doing? And there are data that suggest that hyperglycemia causes reactive oxygen species bursts, at least in adipose tissue. Now, whether, of course, it does in all the other tissues that actually have cancer is still unknown, but we do know that hyperglycemia does that, and the other tissues do take up glucose, so that needs to be explored. Hyperinsulinemia as well causes reactive oxygen species, which then, of course, at least offers the suggestion of a possible mutagen. We don't know that this is true, but if it is, then at least it points in the direction of one way of reducing the incidence of cancer.
Lou, final comment?
So I just raised one additional point that I think is people should be thinking about, which is a lot of people take antioxidants to think because they're protecting themselves from cancer, and almost certainly you can reduce mutational events due to ROS. But by the time you're at my age and Mike Bishop's age, we should be taking oxidants because the tumors, we probably have microtumors already growing, and what limits their ability to grow often is fighting with the ROS that they're generating in the tumor. And we know that in mice with such types of xenografts, that if you give them N-acetylcysteine to keep the tumor better reduced, it grows a lot faster. So in other words, if you're young, sure, take the antioxidants, but once you're at an age where cancers have already starting to grow, switch to oxidants.
Have a cigarette, Lou. Craig? Craig, any last words?
I think that just having this number of people thinking about this number of issues around metabolism again, as we've come through this period where molecular biology and the genomes really revolutionized our ability to build biology, to integrate that back into metabolism is a tremendous opportunity. Ultimately, I agree with Mike. It's going to be the specific causes of cancer, but I think we have not really appreciated the depth to which all these processes that were classically found, receptor tyrosine kinases, the mutations are connected to metabolic pathways, and understanding them. And we now have a body of people that can actually approach that. And I would remind, two questions just came up at the end, or we at least had discussions about. Iron and folate have saved more people in prevention across all of human health than everything else we've talked about over the last century. And yet we still don't have fundamental knowledge of the question. There's lots more. There are many micronutrients we haven't even begun to think about, and there are many individual causes or unique metabolic situations in which cancer arises that haven't even been studied yet. And so there's a wealth of opportunity, and we're going to take, unfortunately, because cancer is, as you say, Greg, 200 or 300 different diseases, it's going to take a long time to get to all the details. But if it keeps us from doing studies and thinking about it, we'll never get anywhere because it would be a daunting challenge. And so we just got to ask the questions that we can. So the examples you used, study oxygen, study what happens to acetate. That matters to actually getting to the understanding of the broader issues of the connections of metabolism to cellular disease, the one we've talked about here, are obesity and cancer.
Steve?
I'll end with a slightly different idea. And kind of through my career, the biggest changes in biological science have been technologically driven, be they started with Ham Smith and Dan Nathan's figuring out restriction enzymes and being able to clone genes, and then PCR. And so technological advancements are just terrific. And I think they're going to be powerful over the next decade for better and better detection. So if the time comes that we can take a blood sample and deduce cancer really, really, really early, I think that's going to be transformative. So whether that constitutes prevention or not, but it really gets back to exactly what Mike started out with, and that is if you don't know the cause, it's really hard to tackle it. So to me, understanding the basis or understanding the cause of the disease is the key point. And that's, as scientists, that's That's what we all try and do.